Thank you. Good morning, everybody. I'm Marissa Hullabar from the Division of ID, and I'm honored to introduce the first ever lecture of the Tompkins-Felco Lectureship, established jointly by the Division of Infectious Disease and Department of Microbiology and Immunology. We founded this annual event to recognize the outstanding contributions of Dr. Lucy Tompkins, who's in the front row here, and the late Dr. Stanley Felco, two giants in the fields of infectious disease and microbiology who call Stanford home. Lucy and Stan exemplified the very best of our profession, a passion for scientific discovery, an unrelenting curiosity for literally everything, and a steadfast commitment to the humans in their academic lives, despite saying often that they were on the side of the microbes. They did all this while approaching every conversation with warmth, a ready smile, and a playful sense of humor. This lectureship was founded by Dr. Upi Singh before she left us to be chair of medicine at Iowa, but is the brainchild of Dr. Julie Parsonet, who's in this audience, someone who we can credit with a lot of great ideas that come out of our division. And in her words, Lucy and Stan not only represent the best of microbiology and infectious diseases research, but also are exemplars of mentorship and leadership at Stanford, in our national professional organizations, and throughout the world. We all love them for their kindness and their ability to bring perspective to the most challenging problems with their humor and wisdom. Lucy and Stan's mentorship is a powerful multiplier of their individual impacts on their fields. Together, they have trained and inspired hundreds of students, postdocs, lab techs, fellows, and we are all extremely proud to be part of their tribe. Despite having this colossal tribe, they all made each of us feel special, heard, and valued as individuals. We all knew that having Lucy or Stan cheering in our corner was a true gift, and one that we did not take for granted. One person I know for sure agrees with me in all these points is our speaker this morning, Dr. Manuel Amieva, another beloved member of our medical community. Manuel is a professor of pediatric infectious diseases and microbiology and immunology here at Stanford. But perhaps most importantly this morning, he is one of Stan and Lucy's most favorite former fellows, and that is a quote from Lucy. Manuel first met Stan in his microbiology course in medical school and credits Stan for inspiring his career in infectious diseases. He was also a postdoc in Stan's lab and participated in joint lab meetings with Lucy's research group. Manuel, like Lucy and Stan, is a microbe enthusiast and a beloved mentor to many. And like Lucy and Stan, he is a person of many interests and talents. Stan flew planes, Lucy loves fly fishing, Manuel wrote children's books, 9 actually in Spanish, published in Mexico before he went to medical school. They're called the Superando series and explore science, discovery, fantasy, and magic. I anticipate your talk today will explore many of those same themes with the characteristic Tompkins, Felco, Amieva warmth and playful sense of humor. So without further ado, please welcome Dr. Manuel Amieva.
Thank you so much, Marissa. It's so nice to be around family. Let me see. I am going to tell you, can you lower the lights a little bit? I am going to tell you stories that are scientific discoveries that my students have made over the past 20 years. And I'm going to try to convince you that we don't really know enough about what the microbes know about us. And you have to use your eyes and your imagination to find these things. And I've been trying to do that thanks to Stan and Lucy. But I want to just start by saying that why are we here? And Marissa just basically said, so I'm just going to summarize some of the things. So We're here because Stanford has people like Stan and Lucy. I came here, I'm an immigrant from Mexico, and really today is very hard for me because I've given many lectures, but this one is probably the most important one I'll ever give. And I had to pinch myself this morning and make sure it hadn't been a dream. So that's like the degree of imposter syndrome that one can feel around people here. But Stan and Lucy are truly giants. And this lectures should try to show you the beauty and wonder of the microbial world that they showed me, as well as the humor. And also that their mentorship is so important. As Marissa said, and we didn't coordinate anything, you know, these two are filled with these two words that I love, both curiosity, but also tempered by humility. I never heard Stan brag about himself. And in fact, I'm just gonna give you one example of this curiosity and humility. In better times, Stanley got all of the awards known to, except for the Nobel Prize, for which he was nominated several times. But he got towards the end of his life, the Presidential Medal of Honor. And he actually went to get it. And here he is with Lucy, with a real president. And what you can't see in this picture is, well, one is that Stan has been crying because to him, this moment meant the recognition of the child of very poor Jewish immigrants being invited to the White House, being given this award by an African-American man, and then bringing his family to see this. But in addition, what you can't see in this picture, and that's often the case with micrographs as well, is that there's hidden stuff. And as you can see, they also decided not just to bring their family, but 3 of Stan's former students. And I don't know if David is here, but Denise is here. And both of them are way more deserving to be up here than me. But I will just be an example. So that was me over there, Forrest Gump. And I'll try to tell a few stories. So why did I name it? You can see a lot by looking. This is a funny thing that Stan used to tell me a lot when when I showed him a micrograph. I thought it was a very wise thing. I actually think that, you know, in Spanish, the word mirar has the Latin root the same as miracle and marvelous. And if you look at something with that sense, you will discover that sense of wonder. But the truth is that Stanley had a favorite philosopher that he was paraphrasing here. And I found out years later that it was Yogi Berra. And Stan was full of wisdom like that came from all kinds of places. I can't tell you some of those things in this audience. But I will try to point out some of the things that he meant. So thinking like a microbe was one of the most important things that he taught me and his students. Is like, put yourself in the evolutionary place of bacteria and think about how they got to where they are and what makes them a pathogen. Pathogens are not just evil things. They have characteristics. I call them personalities. He called them virulence factors. And I teach my fellows that they have to think about what they see in their patient and connect it back to these personalities. In fact, Stan and Lucy were so important about education at Stanford that Stan was still teaching with me and Sharon in the medical school close to when he died. And Lucy continued after that. So bring that to the classroom. And then he always would tell me this, because I thought I was a cell biologist. And he said that pathogens are better cell biologists than us. And indeed, that is the point of this lecture, that we need to study the microbes that live on us because we are just the landscape, and they know more about this landscape than we do because they make experiments, and if they don't do it right, they die. So this encapsulates the story I'm going to tell you today, which you will hopefully understand by the end. But I want you to see it for a moment and imagine what you see. I mean, this is actually the green stuff that looks like a net. It's a tunnel. And inside this tunnel, you see some red stuff. That red stuff is actually bacteria. And this is an epithelial tunnel, or we call it a gland, that is forming deep into your stomach. This is how your stomach regenerates and produces all kinds of things. And those bacteria, what are they doing there? They're crawling into this tube and existing there. This is a piece of human tissue. So I'm going to tell you why I think these particular bacteria there are important. And the theme is really that what makes a pathogen is not just having a virulence factor, but a lot of other things, including where they choose to live and who they choose to interact with. So at the end, you will see that they're actually microscopic hideouts. And I'm going to focus on your stem cells, two things that should be important. You guys all know about Helicobacter pylori in this audience. I think it's very interesting for a lot of reasons. One, it's a human-specific microbe that doesn't have any other place to live, no environmental reservoir. It colonizes about half of the world's population, which, if we call this an infection, it's the most common bacterial infection in humans. It doesn't cause disease in everybody, but in about 15, sometimes 20% of people, it can cause ulcers, which are a disease of the healing of the epithelium. And in fact, now that we know that this is the cause of a lot of the ulcers, we can treat them with antibiotics. So that was kind of a miraculous thing. So finding these things are important clinically. And these people, like this person with a bleeding ulcer, is actually lucky because we can give them antibiotics, we can clear H. pylori, and this ulcer will heal. However, about 1% of the population, and it's a different part of the population, will get gastric cancer. And this is a very advanced gastric cancer, but this happens all the time in countries like where I come from, where people don't have access to care. And when they show up, it's too late. There's nothing to do. And so gastric cancer remains the 5th leading cause of cancer death worldwide. So H. pylori is still killing about 700 to 800,000 people a year. And you guys that work on all of these nasty pathogens, think about that and compare. Because even though this is a very slow burn, in fact, H. pylori is important because it infects us as children, and that's why I can claim to work on it as a pediatrician. It is a pediatric infection almost exclusively, but it's an adult disease. So it spans your whole life. In fact, most people carry the same strain that they acquired in childhood. And I would be remiss if I didn't mention also that a lot of this work finding out epidemiologically how this works comes also from Stanford and from Julie Parsonet in particular. This paper, you know, I was a medical student when she published this, and I've been telling people about this story since then, that they discovered the risk of having this bug in your stomach. Okay, so let's go into a little bit more. Why should we care about H. pylori as biologists? Well, it's a fascinating organism. It's actually not related to the rest of the proteobacteria that you usually think of, like E. coli. It is an epsilon proteobacteria. Now it has a different phylum name, but it's actually related to a lot of extremophiles. Some of them live in the deep ocean, and it just chose the worst place to live in the human body. because it is literally a vat of hydrochloric acid. And everything you learn about the microbiota, eating your food and being happy, doesn't happen in the stomach. In fact, if you are in this place, you will probably be destroyed by the acid. And why would you choose to live here if you could live in the mouth, which is fairly nearby? This is like if you could live in Hawaii, why would you choose to live in the volcano that's erupting next to Hawaii? Well, of course, in biology is because you can, and so we can learn a lot about how microbes adapt themselves even to the nastiest environment in our landscape. And so I'm going to tell you stories with the microscope about how this works. Everything about H. pylori is evolution and adaptation, and it's beautiful. Its beautiful shape, helical, means helical, like a spiral, that's its shape, and has this little tuft of flagella in the back. And you can see that it swims. So I like bacteria because they move, some of them. And you can see that its body is also wiggling as it moves. And if you slow down these movies, you can actually see that the body is also twirling. So think about why a microbe would have this capacity to corkscrew its way through life. And then look at what it also does, which I didn't know until I saw this movie. What just happened? The bacteria just like change directions without turning. And that means it can control its flagella to move like a helicopter or like a boat. These things actually mean something, and I want you to think about it when we get to where it actually lives. Well, I'm also going to tell you a lot of little technical tricks and wrong hypotheses that we've made along the way. And I want you to think about how you would solve some of the questions. So one question we had is, well, how does it live in acid? What happens if we add some acid and see what it does? But we wanted to do it in a way that we could create a gradient of acid. So we made a little injection needle where we can put a little tiny constant pulse of whatever we want into the microscope field. And this is what happened when we put acid. If you look out here on the left-hand side, within a few seconds, the bacteria get out of town. And by 10 seconds, there's almost nobody there. And so Helicobacter hates acid, and it very quickly gets away from it. And the ones on the other side are also seeing that acid, but we've mutated a gene in chemotaxis, so they can't see it or smell it, and so they burn their little noses when they get near the needle. And so that in itself tells you, where does H. pylori live? Well, in the stomach, but the stomach is a very complex place. It doesn't live in the lumen of the stomach. It doesn't eat your food like the rest of your microbiota. It is driven just by the acid to be very close to you and actually to interact with you and feed off of you. So all of the biopsies describe H. pylori basically living in the mucus of the stomach that protects our own stomach from the acid. And that's probably where they're swimming. But also there's bacteria that come and attach to the epithelium. And literally all of the virulence factors that have been discovered have to do with interactions with the epithelial cell. So they even have like needles called type 4 secretion systems. And the most famous bacterial virulence factor in H. pylori is called Kage. And the type 4 secretion system injects things directly into epithelial cells. In fact, that's why I started working on it, because when I joined Stan and Lucy's lab, Lucy had just published, and then 4 other labs across the world had published that the bacteria actually inject this protein, which was associated with cancer, into you with a little microneedle. And I thought that was so outstanding that I had to study. I've been studying it ever since and haven't solved a lot of problems. But here you can see, I started by looking at in the microscope in the library at microscopy images that people had made around the time that H. pylori was discovered, to see if they had seen something interesting. And I saw immediately that the bacteria were here photographed attaching to the surface of the stomach, but they pointed out, G means gutters, that the bacteria are all arranged in the spaces between the epithelial cells, where the epithelial junctions are. I thought that was amazing specificity, and I actually replicated that on a piece of human stomach with antibodies. You see the bacteria sitting all on top of the red junctions between the cells. And so I wanted to study that. And now we have much better tools. So I'll tell you some of the things that our lab has been able to generate as we do this. I had to use cancer cells and transform cells, but now, my students have been able to use organoid technology, so we can actually use human tissues. These are little balls that are derived from stem cells from adult epithelial tissues that grow into these little bubbles. One of the problems is that if you make a little mini stomach like that, you see that the microvilli are facing inside. That means that what would be the inside of the stomach is inside, but there's no entry point. So if you want to infect them, you actually have to poke a hole into them and put bacteria in them, but you make a wound and it's very difficult. So one of the things, and this one was not a wrong hypothesis, but my students and I looked at the literature from another lab at Stanford that was a cell biologist, James Nelson, and he was studying how epithelial cells control their polarity. Polarity means which way it's up and which way it's down. And a lot of it has to do with the interaction with the extracellular matrix. So we argued, well, what if we remove the organoids from these little extracellular matrix bubbles that we grow them in without breaking them up? And would that alter their polarity? And we kept them in this suspension culture. And indeed, you see the green junctions at the top inside Matrigel. You see now they're outside. They flipped in their polarity, and now we have them in suspension so we can infect them. But what's really cool about science is that if you have a good microscope, you can start asking, well, how did it happen? And our hypothesis, and this was another wrong hypothesis, was that organ sitting in Matrigel, Stanley would say, is shuffling around. We thought that it was going to pick up all of its apical proteins and its junctions and move them to the other side, that each cell was going to individually reverse their polarity. And now I'm going to show you how it really happened, which we couldn't have predicted. Watch that movie. That's organ that just came out of the Matrigel. And it'll repeat so that you can see it a couple of times because it's really weird. It actually, these epithelial little balls are acting in concert, and they're like a sock that you pull out of the dryer. The socks are always backwards in the dryer, and it turns itself inside out without having to break the junctions, which is very useful so that we can use it as an infection model. But how do you know that this was actually what was happening? Well, because we can actually make movies and then fix and stain the same organoid in the microscope. That one just got fixed, and we can stain it in situ so nothing moves. And then we can go and look and see, oh yeah, the microvilli are out. So we can prove this. And we can study whether the junctions are intact, and we can study their function, et cetera, et cetera. There are things that these little structures can do that I hadn't even thought about. Like a student asked me, well, can we look at the epithelium from the side and see what the bacteria are doing? And it's like, that's impossible. There's, with a sheet of epithelium, how do you turn it on its side? Well, we can do that with a sphere, because this is a drawing, but here's a movie of an organoid that allows us to see things looking at them from the side. And you can see if we give them bacteria, these organoids, that they whirl around until they find a place to attach. And in this one, we can also fix it right there as it's attaching, and it is attached right at the junction. So within seconds, the microbes are making choices before they actually even attach onto our cells. And this helped us think about, well, if they can sense us, can they actually detect, metabolites that are coming out of us? And my student, Julie Huang, saw that, we can stain them with antibodies against mucus. They produce mucus. Maybe she can collect some of the stuff that's in there and suck it up into that little needle I showed you before and see if the bacteria would recognize that this had been near a human. And I said, no, Julie, that won't work. And believe me, this is the worst wrong hypothesis that you can make. And I've never done it again. And Stan would have agreed, because he always said that the right answer is not to speculate, but to say, do the experiment. And so Julie, of course, did the experiment. And here's what happened. So on the left-hand side, you have this little needle. And within a few seconds, you realize that, oh, they can smell us and they can respond to us. And to me, it makes me feel like they're mosquitoes, and they start itching, right? But then these observations give us the capacity to find the molecular mechanisms. Julie went and mutated the different chemoreceptors and eventually found both the receptor and the ligand for this. And here's an example of what happened when we put pure ligand into the needle. And you'll see in a couple of seconds that there's so much of our response that we didn't even see this little halo. Like, yeah, imagine that. And this turned out to be a metabolite called urea, which H. pylori also destroys with its own enzyme. So it's a long story that I won't tell you. But the microbes are evolving all of these very intricate mechanisms to find us, to figure out where to go, to start interacting with us. Okay, so now that they've gotten to our surface, what happens? Well, I was telling you they make these little injection needles and inject Kage into us. And in my postdoc, I actually found that injection of Kage into polarized epithelial cells would alter the junctions. You see that little jutting red thing under the green is a piece of the junctions that shouldn't be there. And also there's little tiny red dots under some of the bacteria. And we found that it was altering the assembly of the junctions around where the bacteria were attached. And since then, there's been about 4,000 articles on Kage itself. So there's a lot of things that it does, and it's a fascinating molecule because not only is it injected into you, but its function depends on changes that you make to it, like tyrosine for phosphorylating it, which doesn't happen in bacteria. And then it also interacts with all of these things that have to do with cancer, all of these oncogenes and tumor suppressors, and it can break. We found that it can break the junctions if we express it in cells. See that red cell is losing its junctions. It can perturb polarity. It can make the cells confused as to what is up or down, so that the cells actually crawl out of the epithelium. And we and a lot of other people have shown that all of these properties are probably associated with cancer. But that was always bothering me, because like H. pylori will get no benefit if it gives you cancer. If you can think of a benefit, please let me know. But it should mean that this does something for the bug, and nobody had ever looked at it from the perspective of the bug. What does this major virulence factor that's associated with cancer do for the bacteria? And so we started looking at that. And again, when you don't know something, make movies. So we made movies to see what would happen after these two bugs attached to the junctions of an epithelial monolayer. And we spent with my first student all night focusing this thing in the microscope. And we followed these, and we were like surprised that the junctions didn't fall apart. The cells were happy and dividing, and the bacteria were actually making little colonies in the junctions where they attach to. And after about 16 hours, H. pylori divides pretty slowly. We had a little clonal colony of bacteria that were not just attaching to you, they were using you as a place to live. And that changed my whole mind about what H. pylori is doing, because I realized, oh, they're using our epithelial surfaces as a niche for colonization and for survival. And that means that maybe these virulence factors are doing something to make that niche more likely for them to succeed. So Xiumin took then these monolayers and put them in low nutrient media and allowed the bacteria to colonize. And the wild type bacteria grew into these nice microcolonies, but if we mutated Kage, they would roll up into these little balls called coccoids and die. And we could, also culture them, and you saw that the Kage mutant would die, and if we put back the Kage gene, they would come back. So we had a function for this virulence factor for the bug, and then we saw that this was associated with H. pylori causing local perturbations of polarity. So very, very subtle. Underneath these green bacteria, there's a red protein. That protein is actually the transferrin receptor. And you guys know that the transferrin receptor brings iron to the cell, but it never is in the top of the cell because it picks up the transferrin from your blood and gives it to the cell and then goes back to the basolateral side, never to the apical side. And these bacteria are able to bring transferrin and other basolateral molecules to where they're growing. And Shumin showed that they were getting iron from the cell by perturbing local cell polarity. Okay. So where does this happen in vivo, or does it? I mean, I've just shown you a bunch of cell lines. And so my student Josephine Lee started making an animal model to try to study this, and it took her a while to find a strain that would colonize mice efficiently. I could deliver Kage, we could infect babies or adult mice. And then we decided to do 3-dimensional reconstructions of the stomach to see if we could see what I was telling you. And so here's the stomach surface, and there are our H. pylori. We were very happy to see them. They were at the junctions. And then Josephine did something that all students should do, which is to look further, to keep looking. She decided to make thick sections of the stomach and look through the whole mucosa. And when she did that, there's a bacteria at the top in the mucus, and you see some attaching at the surface. Now we're looking at it from the side. And when she looked down into the glands, then she found this other population of bacteria that we didn't know existed. And these bacteria are very happily dividing in your glands, and they're very well organized, at least in these mouse. And the region where they are turned out to be very special. I don't know if you guys know what this region is, but even with just this simple stain, this DNA stain in blue, we can see that they are interacting with your progenitor cells, the cells that are actually dividing. And we can just look at the cells that are in mitosis by staining the DNA, and that's the region where they are. And you can stain it with antibodies. They're in white. And you can see that the bacteria like to live right on top of your dividing cells. Well, now that was exciting because now we have a bug that can manipulate your epithelial cells, but it's actually in the place where your epithelial cells are long-lived and dividing. And that could have something to do with cancer. But we didn't actually know if this happened in humans. So we actually collaborated with friends and scientists in Mexico that sent us infected large pieces of tissue from patients that were undergoing bariatric surgery. And in Mexico, the H. pylori incidence is over 70%. So we could find patients that were infected and asymptomatic and see if we could find the bacteria where we're telling you. And this is what a pathologist usually sees, although this is prettier because we aligned it very nicely. This is a 5-micron section, but we had big chunks of tissue so we could make 100-micron deep tissues and know exactly where we were. And taking that exact same slide, if you try to look for the bacteria, this is what you get for diagnosis. You see these little squiggles up there. And actually, that's how it was discovered by Barry Marshall and Robin with that kind of data. But it's really hard to see. Those are the bugs sitting on the epithelium. Well, if you take this exact same tissue and you look at it with 3D confocal microscopy, you can see that, there's a lot of bacteria swimming in the mucus at the top. If you turn the top, you see that there's bacteria attaching to the junctions of the surface of the stomach. But if you look deep in the glands, you find these populations of bacteria inside the human stomach that also are associated with mitotic cells that you can stain. So we were really excited that the stuff that we were seeing in the mouse and that we had seen in cell culture, we could recapitulate to some extent in the human, but there's so much that we don't know about this that I can tell you later. One thing is that we also found signal of H. pylori way at the bottom. This red thing is the end of the stomach. If you have a gland, that's where that signal is coming from. So we wondered, is H. pylori actually affecting the stem cells, which live way at the bottom? Because those are even more important. And cancer, we think, may be evolved from stem cells gone awry. So we looked in the mouse model. And the nice thing is that you can get mice that have green stem cells, and we infected them. And Michael Segal did this work in my lab, and we found indeed that there were populations, about 30% of the gland bases had little colonies of H. pylori living right on top of the stem cells. And then he did a very simple experiment. He infected a mouse for a month, and that mouse, after a month, had twice the number of stem cells. And just with this mouse is asymptomatic. It doesn't have cancer. There's nothing bad going on. But now it has twice the number of stem cells with bacteria that are making them proliferate more. And that in itself increases the risk of cancer in other studies. So, oh, and by the way, the stem cells know that they've been infected by 2 weeks of infection, which is the earliest we look. You don't have to wait a month when there's already inflammation and all kinds of other changes. And the stem cells actually start to express genes that this is in situ hybridization. And I mentioned this one, olefin 4, because it's going to come out at the end of the story. This was in a mouse infected for a month. We also made organoids from these infected mice. And the organoids cannot remember that they were in an infected mouse, because they made more organoids and they grew faster, even after you took them out of the stomach. So the stem cells have some kind of memory of having been near H. pylori. And we also wanted to know whether this was due to the proximity of the bacteria in the infected glands or just infection in general. That was a harder question. And I'm just gonna show you what I think was the best experiment, because it was done by, because of stuff that Michael Howard, who's now an assistant professor here at Stanford, working on something completely different. But he found this molecule in H. pylori that you see there at the tips of the bacteria that is a regulator of chemotaxis, but is only found in this phylum that H. pylori comes from. And this protein, which he named KPEP, when we infected mice, they colonize just as well as the wild type, which is interesting. They don't really need it. But then when we looked at the stomachs, they were all sitting in the top in the mucus, and they were not going into the glands. So this was a great tool where we could ask, well, what is the significance of the bacteria not being able to colonize the gland niche? And we asked that from the side of the host and later from the side of the bug. From the side of the host, we found that if we infected mice for 6 months, you can see how inflamed and hyperplastic the gland looks after 6 months. But mice infected with the KPEP mutant did not undergo these processes. So even though the bacteria are there, your stomach doesn't care. And so now, for the last few minutes, I want to tell you about what is in it for the bug, and then some new stuff, because I know that Stanley would have liked to see new technology. So Connie wanted to ask, what's in it for the bugs? Like, why do they want to go there? And we had this clue that was really cool. If you take animals and infected with this mutant that doesn't go in the glands, or the wild type, you see those dots up there, they're equal. They don't really care. But if you pre-mix them before you give them to the mouse, and then you wait and you see who wins, the wild type completely outcompetes the mutant. So there's no mutant left in those stomachs. It was very dramatic. It means that there's some benefit of having that gene that completely overcomes the capacity of that mutant to colonize. So we started with this project where we made identical twin sisters. One is red and one is green. They are colorblind, so they can't see each other. And they also are otherwise identical. And they infect the mice the same numbers, they go into the glands, they do all of that in single infections. When we mix them, they infect the same mouse at the same level. And when we started looking at them, we see them in the mucus, they're mixed. All of that was going well. We see them in the glands, yeah, they were mixed as we expected. They're coming in and out of the glands. And then we looked further. And then we started seeing glands that had more green than red, more red than green, and glands that had only red or only green. And that's when I started to worry. How is it possible that these bacteria that we mix at about 10 million in a mixture are deconvolving themselves? And so I asked Connie to please tell me, what is the distribution? Are there more mixed and those are very rare, or is it the other way around? And she went and did a lot of quantitation and came back and said, oh, there's more single occupancy glands than mixtures. I'm like, oh my God. Well, does that mean that these are founders and that they're mixing? And why don't you count more glands? And so she counted 1,000 glands and she came back with the same result. And so then we again had to figure out ways of trying to figure out what's going on. So we started looking at the location of these colonies in the stomach without disturbing the anatomy. And here, look at this picture. It's a movie of going through a whole strip of glands in a mouse stomach. And you see here the bacteria in red. And we're moving through this. And then all of a sudden you see red, 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 and now they're mixed. And now then at the end, they're green. And so when you actually look at where the bacteria are colonizing the stomach, you remove the signal from the stomach, you can measure things. There's the same amount of bacteria, but they're clearly organized into different regions. So they're populations. They're not just randomly colonizing glands. And we did this with many mice, et cetera. And in fact, we even used a technique that was developed also at Stanford for clarifying brains. And we clarified a stomach, turned it into a little glass thing. And we made stomachs that were flat so we can look clearly at the glands. And so here are like 12,000 glands. And we can get really close to these and see the glands and count them. And we can see the bacteria in them. So this is a fancy 3D reconstruction of this, where you can see the glands going, the bacteria going into the glands and congregating there in the precursor zone. But we can also take a bird's eye view of this. We can go back out and see where are these populations of red and green bacteria. And there they are. They're forming islands, population islands that colonize the stomach as independent populations. And where we see them mixing is where these two things meet. And this was really, again, changed my mind about how is an organ colonized by bacteria? What is the dynamics of population change in our bodies? We asked, well, are they gonna ever mix? And we let them colonize for a long time, and these islands grew, but they didn't mix. So we asked, a professor of ecology to help us model this. And indeed, it looks like these are founders that are growing as an island. And why am I telling you all this? This is a medical group. Well, because this actually has lots of practical implications. If the microbes are colonizing special places in your stomach and then not letting go, Stan would call that squatter's rights. Then what happens if we first infect the stomach with one bug and then infect with the other twin sister? What will happen then to the second one? Can you guys make that, think about that for a second? Well, here's if we infect them in a mixed infection. I told you they infect equally. But if we do sequential infections, the second one cannot colonize. So that means that going into the glands creates what we call colonization resistance, which is a fancy word for squatters' rights. And this happens really fast. After 3 days, you can still see these mixed colonies. And after 5 days, all the glands are the color of the second one. Okay, so what happens if we then first colonize the glands with the mutant that can't enter the glands? What do you think is going to happen? If we then wait and give this guy a chance, then add the wild type. Well, again, this is what happens with the wild type versus wild type. If we give the mutant first, and at the end, they're all the first one. If we give the mutant first, the wild type actually takes over and eventually kicks out the mutant. So this is all to tell you that H. pylori has evolved and discovered these hideouts where it has an advantage and it's messing with your precursors and stem cells. The last couple of minutes, I just want to tell you some stuff that's going on in the lab. And it's also because this represents an incredible collaboration with people that are bringing medicine and pediatrics together as well. So this is a team of very talented gastroenterologists, medical oncologists, and omics experts that brought us into their fold. They had discovered a signature in people that are at risk of developing cancer. They have advanced metaplasia, intestinal metaplasia of the stomach, and they can find the stem cells of these metaplastic regions have markers of intestinal stem cells, which is a new marker for who's at risk. They just published this. And they asked us, could we collaborate and turn this into an investigational platform where we can take these tissues and turn them into a live biobank? And so that's what we've been doing. And Yulim and Lorena from my lab were able to make these organoids just from single biopsies, from little tiny pieces, and grow them directly from the patients. Thank you to the patients. And there's an organoid, a new little line coming up in 9 days. And now we have about 60 patients lines that we've generated. And we're discovering that indeed the metaplastic organoids maintain these olefin 4 positive, intestinal-like stem cells. And that's kind of where we're at with that. And we're starting to ask questions. Well, now that we have these lines from different people, different parts of the stomach, can we start asking questions about how are the stem cells changed or do they matter? And so one simple experiment that Yulim did, is she started with either precursor organoids that I've been talking to you about, but we can also differentiate them into the top of the gland, which produces lots of mucus, so surface type organoids, and infected them with H. pylori. And most of the responses that she sees are from the precursors, as if the guys that are already differentiated don't care. And then she found out that these molecules are a lot of inflammatory molecules, that the epithelium also has receptors for the cytokines that are produced. And that's when I started getting a little bit interested in immunology, because I like the epithelium, but people don't realize that it's also an immunological organ. And we have signatures of cytokine receptors expressed in these organoids. So Lorena started sprinkling cytokines, and the organoids respond. And here's like some of these organoids getting really big and strange in their shape, just with a little bit of IL-17. I'm almost done. I just can't stop without showing you the last story. Yulim also is turning these cultures into long-term cultures. She can put them in a transwell and then remove the media, which puts air on the top. And for some reason that helps them stay there. They start making a lot of mucus, so people call them mucosoids, but they also have lots of stem cells there. And the ones from the metaplastic tissues express metaplastic stem cells with that marker, the olefin 4, in those little dips. It's as if they know that the dip is maybe like a little gland. And then when we infect with H. pylori, you see that the H. pylori also know that there's little tiny glands in these things, and they like to congregate there. So now we have a system where we can start exploring this also in disease tissue. Okay, last story, really quick. Because Stan would have loved this. Because this student, PhD student from Germany, looked me up and sent me a message, say, I'm a marine biologist and I study the epsilon proteobacteria in the deep ocean. And I would like to come and work in your lab because I want to learn something where I can do experiments, because we drag these things with a submarine robot and then they come out and we can't keep them alive. And so he came to Stanford. Those are the epsilon proteobacteria in the deep ocean. And he's now learning to be a microbe hunter out of the water. And I was interested in this because when he looked at his deep sea mussels, they have these gland bacteria living in them that he could find by in situ hybridization. And he was using this very special technique called metabolic imaging, where I'll show you how it works. You shine a laser on the tissue, and without adding any stain, without antibodies, without probes, you can see a picture. And Stan would have loved that one. So he takes sections of the organoids and he puts them in this matrix, and he takes them to this really fancy microscope that the pathology department, Michelangelo, who's like Michelangelo, has and has allowed Benedict to use. And then he can scan the tissue with this laser, and the bugs here are labeled with GFP, so they're in green. And all of those peaks that you're seeing there from the monitor are individual molecules. So in the end, he's left with thousands of little images that actually have different metabolites. So you see these organisms with different distributions of metabolites. And so now we can start asking, can we see the bugs? Can we see what they're doing? And indeed, he's using spatial correlation, teaching the computer to find metabolites that are associated with the bugs. So here's a metabolite that's shared by the host and the bug. Here's a metabolite that's mostly in the bug. Here's a metabolite that's only in some microcolonies. Here's a metabolite that's only in the microcolonies. And I said, stop, Benedict. Find something that you can make mutants in. And so right now he's doing that. We are very grateful that another student joined our lab, and Kate is an expert molecular biologist. So she's helping Benedict mutate the enzymes that lead to this triangle. This triangle is called cyclopropanation, and it only happens in certain bacteria. Humans can't do it. So the cool thing is that to test this mutant, Benedict puts a little dots of bacteria in a slide and takes it to this fancy microscope, and there's the wild type that has made that triangle lipid, and the one that he made the mutant in doesn't have it. So Kate is still happy. Now because we have red and green bacteria, they're putting it into this background so we can watch them as they attach to your epithelium. And they could be metaplastic epithelium, and we can watch how they grow and measure that. And we can also see already that they attach at the junctions, and that here the green is a junctional protein. You see this little patch of green that shouldn't be there. That's where the bugs live, are created by infecting this organoid. So I've been able to recapitulate the stuff where I started in using humans. All right, I'm done. But I just want to acknowledge people. I've been acknowledging our students and our collaborators. But actually, I just want to end with the most important person here, which is Lucy. And I am so honored to be able to say this in front of all of you. You can see she is opening her arms to students that she and Stan had invited to their home in Montana, and gathering us and That is what Lucy does. Thank you. And I'm sorry that I didn't leave a lot of room for questions.
That was wonderful. Maybe some time for one or two questions. Questions from the field here. Yes.
That was great. I was thinking about your little dips in the epithelium, and you remember that there's some force sensing in the apical surface of your organoids. So I was wondering if they're sensing, if there's some kind of force sensing going on as in the dips.
Yes, the answer is yes. There's actually people, we just collaborated with a group that actually creates scaffolds made using photolithography. They basically paint the extracellular matrix structures, and then they put the organoid cells. And so these cells will themselves sense that they're in a gland and turn and become more immature, whereas the ones that are on the top will be more mature. And so the cells definitely are sensing their mechanical properties, and that's part of the reason they know that they are a stem cell.
That was just such a beautiful talk, and it was such a great honor to Lucy and Stan, who inspired a lot of us to work on Helicobacter, and just really, really beautiful. I just want to ask about CAG-A negative strains, because you talked a lot about CAG-A positive strains, and a lot of strains in the world don't have that, and people are not infected with any CAG-A positive strains. And so I'm just wondering how they fit into this picture.
It's a good question. I've been asked that question all my life, and the answer is I don't know. I mean, you know that H. pylori is so diverse and heterogeneous that we know very, very little about the CAG-A negative strains. We've been trying to study CAG-A mostly, and so do they have other things that function like CAG-A? Do they not need them? The way that I think now about H. pylori, and I think that this is, kind of hand waving, but there's a beautiful paper by Correa, the guy that described the Correa cascade towards cancer, where they looked at people in this population he follows in Colombia that all have H. pylori, but the ones in the mountains have a really high rate of cancer. The ones near the ocean almost never get cancer. And so they started doing they've looked at everything, their diet, their behavior, their genetics. And what has panned out the most is that when they biopsied people and then cultured their H. pylori and then looked at genomically at the phylogeny of the person's genetics and the bacterial phylogeny, they found that the people that were getting more disease are the people that had a mismatch, that they were infected, they were Afro-American infected with a Spanish H. pylori, or they were, Indian infected with African H. pylori. And there's this mismatch in the evolution of the microbe and the host, which is very, very difficult to boil down to a single gene, that leads to bad outcomes. So that's something that's an example of this intricacy that I really like. But I don't know if you compare, there's something about CAG-A that is important, because otherwise they would have all lost it. Because it's a very big set of genes. It takes a lot of energy to make this type 4 machinery. And so there are some situations in which they really need it.
Manny, it was a wonderful talk. Over here, Michelle. I'm going to ask a very naive question as a clinician, but is there any
substance or protein or way to interfere with the actual receptor for H. pylori onto the intestinal epithelial.
So
So that we don't have to give this horrible cocktail of antibiotics
Yeah.
that makes everyone sick.
yeah, so thank you. That's a really great question because again, I think Stanley would say that the basic biology is what allows us to find alternatives. I mean, when we use antibiotics as infectious diseases, we're throwing little atomic bombs throughout our body, right? And we're thinking we're killing H. pylori, but H. pylori ends up surviving, and then we wiped out a lot of these beneficial microbes in your gut. So it's turning out that H. pylori is really difficult to eradicate. And you need 2, 3, 4 antibiotics for 2 weeks, proton pump inhibitor, and still you get, failures. So how do we get around that? Well, one of the theories that I have is that it's these hideouts where they recrudesce, and we have a little bit of data about that. So we have to understand how do we draw them out, and maybe then if you draw them out, you can treat them for 3 days, just like any other infection. But it also makes me think or imagine that microbes that are mucosal colonizers, not just H. pylori, but Staph aureus, all of these other ones that we deal with, they have really special ways to survive and persist because like you can give tons of antibiotics and they still there. So I think we need to understand colonization niches, not just how to clear an infection so that we can eradicate things or come up with things that can perturb that.
Maybe we'll do one more and then we'll close. Thank you.
Yeah, amazing talk. Thanks, Manuel. Can you use the squatter's right phenomenon to actually sort of immunize people with strains that are CAG-A negative or other low oncogenicity associated strains?
Yeah, that's a great question. So people have talked about, there's good H. pylori and bad H. pylori. Why not we just give everybody a good H. pylori and be done with it? And I think people have considered that, but there's so much we don't know. In fact, H. pylori is probably in most everybody, but a few children, not just a commensal, but even a symbiont. It probably provides benefit, and I didn't have time to talk about all of that. But it is one of these microbes that really educates your immune system and makes it tolerant. And if we just treat children with antibiotics and get rid of H. pylori, we're seeing the consequence of that is you have more autoimmune diseases. So figuring out what bug to put in is not going to be easy, and it might be hard to get it past the FDA. But I think it's a brilliant idea to try to create microbes. In fact, you would have to give it very early in life because otherwise the natural one from the mom is the one that's gonna colonize. But yes, that is, I think, possible.
We don't usually have a packed room, and we certainly never have a packed room at 9:06. That says a lot about the importance of this lectureship, honoring Dr. Falco and Dr. Thompson. Thank you, Dr. Amieva, for doing such an amazing job. Thank you, everybody.
The preceding program is copyrighted by the Board of Trustees of the Leland Stanford Junior University. Please visit us at med.stanford.edu.